Okay, Lucas.
Hello all. Thank you for being here after this period, this break that we took, right? So it's really good to have you all here. So today it's a pleasure to introduce my colleague and friend, Andre Naranjo. He's a current NSF Biological Collection postdoctoral fellow working on patterns of endemism, phylogenetic, and functional trait diversity in Hispaniola mountain systems here at the Florida Museum of Natural History with Lucas Majur. And he's originally from Miami and received his PhD in botany here as well in the University of Florida last year, right? Working with Pam Sotis as his advisor. So Andre, thank you so much for accepting and we are all really excited to hear all your amazing work. So whenever you're ready.
Great. Well, thank you, Lucas, for the introduction. Also, a shout out to Agnes for having invited me in the first place. I honestly thought no one would look at my botany talk, so I was very, very excited, very happy, and very pleased that people, especially melastome people, were taking interest into what I was doing, which was essentially just using melastomes as a kind of like a preliminary model to get a feel for patterns of biodiversity and endemism in Hispaniola. But for this presentation, I think I wanted to go on a slightly larger breadth and look at Caribbean melastomes, in particular Myconii, and use those to kind of tease apart, patterns of phylogenetic diversity, of endemism, and also to kind of do something that I really love, which is divergence time estimation and biogeographic analysis. And using methods I had never used before on a relatively large data set. So thank you again for having me here. So I wanted to get started off. Let's see, let me see if this works. There we go. On just a very quick overview of kind of like some of the main melastome groups that we see in the Caribbean. And the Caribbean and its constituent habitats are what I would consider a living museum of biodiversity, in particular for New World melastomes. Melastomes in the region, they largely belong to the tribes Blakeae, Henriettiae, and Myconiae. Myconiae, the latter, being the most diverse tribe of Melastomataceae with 30 genera and around 2,200 species. That tribe, which is this one over here on the left picture, I believe that's a Myconia, is characterized by fleshy fruits and partially the totally inferior ovaries. So to give you a brief overview, and I'll touch on each one of these aims and the methods and results subsequently. I was really aiming to answer the following questions. First, trying to tease apart the natural history of Myconiae. How old are the constituent clades, or at least the clades of interest, and what were their ancestral areas? Then looking, using the phylogeny to understand relationships across the different mountain systems of the Antilles and surrounding areas. So were these relationships over-dispersed or clustered? And then lastly, focusing a little bit more on Hispaniola in particular, was understanding the roles that neo and paleoendemism played in shaping the patterns of endemic species that we see across different mountain ranges on the island. So first off, to just jump right in, the first aim was to build a phylogeny to better understand the divergence time and ancestral areas of this hyperdiverse clade. And once we, in order to do any subsequent analyses, we really had to work first on building a dated phylogeny and to better understand patterns of phylogenetic diversity and endemism subsequently. So to build that, we obtained sequence data for ETS, ITS, the ACD PSL spacer region, and PSBK from DNA extractions to produce a phylogeny for all the species, or for most of the species in the group. These genes were chosen in particular because of their utility in teasing apart relationships in clades that might be the product of rapid radiations. We then concatenated and partitioned the resulting DNA matrix and ran a maximum likelihood method implemented in RAxML, as many people might be familiar with. We then utilized a penalized likelihood approach in TreePL for dating in conjunction with our concatenated and partitioned maximum likelihood topology. Our confidence intervals, so many people that have worked with TreePL in the past know that usually you give it a tree, it runs this penalized maximum likelihood algorithm and gives you a date for a node. It doesn't give you a confidence interval kind of like the way BEAST does, because this penalized maximum likelihood doesn't give you a distribution of trees. So what we did was we calculated the confidence intervals by accounting for variation in branch length. We generated 100 bootstrap replicates from the multiple sequence alignment with RAxML. We constrained the topology to the concatenated tree, but allowed for varying branch lengths. And then we subsequently ran TreePL on the 100 replicates. And then node ages and statistics were summarized using tree annotator. So in the end, BEAST ended up coming in useful. We constrained the most recent common ancestor of Henrietta and Myconi having occurred sometime between 38 million to 58 million years ago. Then, because of the size of our tree, several hundred terminals, DEC likelihood model was just not computationally feasible. We were attempting to run it for over 300 hours with almost 100 gigabytes of RAM and nothing. The jobs were constantly failing. So we therefore decided to use the dated TreePL topology and in conjunction with the C++ program called Bay Area that uses stochastic mapping in conjunction with a Markov chain Monte Carlo to sample range evolution histories that were consistent with ranges observed in extant study taxa. So Bay Area is different, is kind of like DEC. It adopts a DEC-like approach, but it also allows for a wide range of processes, such as within area speciation, range expansion, which is dispersal to a new area, range contraction, which is extinction in an area that was previously in. And so we ran this analysis for 50 million generations, and we removed the first 20% as burn-in. So we recognized 14 biogeographic areas in the analysis based off of major mountain ranges in the literature that were also identified as phytogeographic regions of importance in the Caribbean basin. And so the 14 areas we recognize in order, and I don't know if you can see my cursor right now, but I'll just quickly go over them, are the Massif de la Haute, the Massif de la Selle and the Sierra de Bauruco, the Massif du Nord and Cordillera Central along central Hispaniola, the Sierra de Neiva, which is just north of Bauruco, Moatoa Baracoa and the Sierra de Cristal in eastern Cuba, Pina del Rio, which is a sub-mountainous region, but it has mountain-like features, the Trinidad Mountains, the Sierra Maestra, also in eastern Cuba, then the Cordillera Septentrional and Samaná, which is in eastern Hispaniola, Jamaica, Puerto Rico, and then a large assortment of Mexico, Central America, North America, which I'll mostly refer to as Central America for simplicity's sake because it's in the map, South America, and then the Lesser Antilles. And you'll see this map in each of the subsequent topologies that when we cover ancestral areas. So this is our initial resulting topology from TreePL with the 95% confidence intervals for each node dated. So it's a bit messy, obviously. So let's focus in particularly on the nodes of interest that we were wanting to look at. And so first off, we have the in-group node. So the out group is Henriette, and in-group is pretty much everybody else. So this in-group node includes both the Myconi and the so-called Ariocnemi. They were reconstructed as having occurred somewhere in Central or South America about 30 million years ago during the Oligocene. Looking at the ancestral node for Myconi, it was reconstructed as having occurred again also in either Central or South America, approximately 15 million years ago during the Miocene. So within the Myconi, I'm going to change up the topology, but to make it a little bit easier to visualize where these clades are, we looked at the Chenopleura clade, which can be easily kind of seen on this topology with its shorter branch lengths relative to the surrounding clades. This is a mostly Hispaniolan and Cuban clade, and its ancestor was reconstructed as having occurred in South America about 2.7 million years ago during the very early Pleistocene to very late Pliocene. Next up was the Macranium clade, an even shorter branch length group made up of many seemingly recently diverged species. This is a largely Hispaniolan clade with many members endemic exclusively to the Massif de la Haute in Western Haiti. Its ancestor was reconstructed as having occurred in Central America, approximately 6 million years ago during the late Miocene to early Pliocene. We then looked at the larger so-called Caribbean clade, which is made up of members of the genera, or at least the classically described genera, Pachyanthes, Caryanthus, Tetrazygia, Calicogonium, Osea, and Myconia. And if I left anyone out, please let me know. It's made up of larger branched groups that than the previously highlighted clades, as you can tell. Extant species are almost exclusively endemic to the Antilles, but interestingly, the clade's ancestor was reconstructed as having occurred in either Western Cuba, Pinar del Rio region, or Central America, approximately 9.6 million years ago during the late Miocene. And then the last clade within the Myconi that we wanted to look at was the Brevissima subclade of Sagria. It's nested within the larger Sagria clade, and it's made up of, I believe, 29 species that mostly belong to the genera Clydemia and Ossaea. And this clade, which is endemic to the Greater Antilles, had its ancestors reconstructed as having occurred in the Massif de la Haute in Western Haiti, approximately 3.3 million years ago during the late Pliocene. So moving on from there, the second aim was to look at relationships across the communities. So there are multiple nuclei for endemism in the Caribbean island biodiversity hotspot, and using sort of recently developed methods for better understanding phylogenetics at a spatial scale, it's now also possible to evaluate biodiversity by combining both locality data from museum collections and DNA-based phylogenies. So using this comprehensive phylogeny of Caribbean melastomes, in particular Caribbean Myconi, we hoped to tease apart spatial phylogenetic patterns across Hispaniolan mountains and also across neighboring islands as well. So using the topology that we built for our dating and biogeographic analysis, we calculated measures such as phylogenetic diversity, mean pairwise distance, and mean nearest taxon distance, which I'll touch on the definitions of each in a little bit, so that we could then subsequently compare diversity metrics kind of give some background as to what we were thinking in terms of predictions. Is the underlying principle of island biogeography states that species richness must result from in situ diversification in larger areas. So it's unclear exactly how these two competing processes affect phylogenetic diversity, particularly in the context of vascular plants. Studies have sort of uncovered that ecological processes such as colonization from continents are the main contributors to phylogenetic diversity and richness, not evolutionary processes like diversification in situ. So our predictions were, I mean, one might expect the mountains, in particular the mountains of Hispaniola, to show similar patterns because of their location on a relatively large tropical island. But it differs in that floristic studies of its flora show certain lineages, and in particular Myconiae, as being overrepresented in the endemic flora, which is an indication that in situ diversification might be occurring. So we predicted that these mountain communities, in particular those in Hispaniola, are phylogenetically clustered, and that species richness is a product of mostly in situ diversification and not so much frequent colonization from larger continental landmasses. So here are our results for our diversity metrics for all these different mountain ranges. And I have the legend off to the side. It's the same biogeographic areas that I mentioned before. So in this case, we calculated phylogenetic diversity, particular Fates phylogenetic diversity, which refers to the sum of the lengths of the branches found in a community. The highest values for this metric were found in the Massif de la Haute, which is a well-described endemism hotspot, followed by the Massif de la Selle, Bauruco, which we kind of put together because they are kind of the same mountain range, and the Massif du Nord Cordillera Central, all of these which were located on Hispaniola. And I'll mention that I have I have the Hispaniolan areas in the boxed region to just kind of like give you a reference for how Hispaniolan measures of diversity compared to the surrounding regions. We then looked at MPD, mean pairwise distance. So this measure calculates the mean phylogenetic distance, which is obviously in the form of branch lengths. So it essentially compares the branch lengths among all pairs of species within a certain community. And so MPD, for the most part, gave us results that were similar to the phylogenetic diversity metric, except that here it identified the Sierra de Neiva in central Hispaniola as being the most diverse, followed by the Massif de la Haute, the Sierra de Bauruco, and the Cordillera Central. And then what I think is probably the most interesting one is the mean nearest taxon distance, which has a pretty self-explanatory name. Essentially means it's the distance between each species within a community and its closest relative. So this one showed that Sierra Neiva was as diverse as the MPD results had shown, followed by the Lesser Antilles, and then Central and South America, kind of tied for third place. So we see a complete reversal in the diversity metrics scoring here, because the least diverse area using MNTD was the Massif de la Haute. This is a clear result of the fact that the community phylogeny for the Massif de la Haute is made up of very compact, short branch lengths, indicating that it's a community made up of relatively closely related taxa in comparison to these other regions, which have more of an assortment of species that are perhaps not as closely related and might be, in fact, the product of not in situ diversification, but rather colonization from either islands or large continental masses. So let's focus a little bit now more specifically on the most topologically heterogeneous island of the Caribbean, which is Hispaniola. It's an island that also hosts a very large chunk of melastome diversity in the New World. So one very important goal of my project is to present diversity metrics to better inform land use decision in these ranges and in these islands in these countries. So understanding the patterns of biodiversity, particularly the patterns of endemism, is critical for conservation, given the very rapid habitat loss in Hispaniola and other islands in the Caribbean. And sadly, the effects of human-induced climatic change in the form of stronger droughts, heavier downpours, and stronger hurricanes, as Hispaniola this year has already had the sad reality of having to deal with 3 different tropical systems in a matter of a month. So in order to calculate endemism, endemism is usually assessed by comparing species ranges across a region and then counting a species number to give you a species richness score. But analyses implemented in Biodiverse can help distinguish against different types of different centers of endemism in ways that are informative for understanding the ecology and the evolution of a mountain range in a region. So for this, we incorporated locality data with our phylogeny to then use Biodiverse to run phylogenetic endemism analyses and a so-called categorical analysis of neo and paleo endemism, otherwise known as canopy, to understand 4 different meaningful categories of endemism. So those 4 categories of endemism are defined as follows. Areas of neo-endemism, which are made up of rare short branch species. Areas of paleo-endemism, which is made up of rare long branch species. Centers of mixed endemism, which include a mix of both short and long branches. And areas of mixed endemism that are referred to as areas of super endemism. So our goal is to essentially determine if there's an overrepresentation of range-restricted long or short branch species in a selected location, as compared to what would be expected if the same number of taxa had been selected by randomly from the tree. From this, we would then further compare geographic grid cells identified as significant, and we run a canopy analysis to identify areas of endemism that share similar sections of the phylogeny. So given past studies of endemic diversity in the region, we expect much of the taxa to occur in areas labeled as neo-endemic due to the presence of large clades that have recent diversification and radiation events in the region. So another explanation for this pattern might be ecological. The results of habitat filtering based on phylogenetically conserved traits that then result in close relatives of co-occurring species in the same communities, leading to these phylogenetically clustered habitats, such as in the Massif de la Haute, that arose as a result of neo-endemism. So here are the results for just the phylogenetic endemism analyses for melastomes. And we uncovered hotspots of endemism in the areas seen in red. In particular, the Massif de la Haute, La Selle, and Bauruco, with a little gap in between. And in northern areas of the Cordillera Central and Massif du Nord have these so-called endemism hotspots. But this alone doesn't mean much. So we wanted to look at relative phylogenetic endemism to see if these patterns hold up when pulled from random from the tree. And revealed similar patterns as well. Again, hotspots of endemism were seen in the areas in red, in particular, areas of La Haute, La Selle, and a particularly large hotspot in eastern Bauruco, northern areas of the Cordillera Central as well, and areas of Samaná and the Cordillera Septentrional were recovered as having high levels of relative phylogenetic endemism. But perhaps the most interesting part for me was looking at what sort of patterns of neo, paleo, mixed, or super endemism were we going to see with our canopy results. And we didn't see any areas of mixed or super endemism, but we did uncover several different hotspots that were of interest. And that kind of gave a little bit of credence to our initial hypothesis. We were able to uncover these neo-endemism hotspots, in particular in the Massif de la Haute and in Eastern Bauruco, among other areas, such as Sierra Neiva as well. While we uncovered a fairly cohesive area of paleo-endemism in the Cordillera Central. And this matches up well with our mean nearest taxon distance metric results, that showed that shorter branch communities like La Haute and Eastern Bauruco were recovered as areas of more recent endemism in comparison to Cordillera Central, which seems to have a community made up not so much of in situ diversification or speciation that occurred in the mountain range, but rather probably a kind of a biodiversity catching net of catching species via long distance dispersals and probably birds as well, visiting essentially the highest mountain range in the Caribbean. And so with that, I would like to thank the University of Florida, National Science Foundation, the Florida Museum, and everyone at the Mazur Lab for all of their help. And thank you for listening, and thank you for coming.
Thank you so much, Andre. That's very, very interesting. And I think it will be great to see how this compares to other clades and other completely different families.
Yeah.
You guys are looking at. So we open the floor now for questions. We have plenty of time. I have like 50 questions, but I don't want to hug all the time. But I'm gonna start with one, and it's if we look at neotropical melastomes, about 58% have berries and 42% have capsules. But then when we go to the Caribbean ones, it's almost all, I mean, not all, but it's less than 20 species are capsular in the Antilles out of 400 and change. So I want to know what you think about that. And then the other question I'm going to ask right away is, for these areas of endemism, high endemism, they're obviously related to elevation, like which is happens in the Andes, happens almost anywhere we looked at. But there are some that seem to be very near the coast, and in some of those. And I wonder, do you know anything specific about those locations? Are those like serpentine soils or nickel mines, or you have a little bit more idea about those not high elevation but high endemic areas?
Well, I'll touch on the last question first. With regards to specific soil types, I don't think there was anything I read that indicated that these hotspots were related to some sort of special edaphic condition. It's kind of interesting because, I mean, the grid cell size is of a certain, you're only allowed. So the grid cells I based off of were based off of something in the literature I read with regards to the grid size should only be as large as the smallest range. And so again, some of these are very limited, and some of these species are only found on one site on a mountainside, right? So this is a methodological limitation, I guess, that we're seeing here. It would be amazing to get less coarse, more high definition resolution for these localities. Locality data for some of these is a little bit hard to come by, and subsequently inputting into canopy and biodiverse has its limitations. But I expect that the lower elevation hotspots are more a product of the methods and not so much a product of actual hyperdiversity in those regions or endemism. And then your first question, it's the a small little ecological part of my brain says birds, right? If there is a clear shift to a certain fruit type, such as from capsules to berries, indicates to me that there's some sort of reward for dispersing the seeds, right? In this case, it would be a fruit, a fleshy fruit. I'm going to be touching on this a little bit more when I deal with a functional trait aspect of my postdoctoral project. I'm looking at fruit types and other functional traits for several hyperdiverse clades in Hispaniola, one of them being the MELASTOMES. In addition, Pilea, Peperomia, other very, very diverse groups that are found on these mountains. And I hope by then to be able to better tease apart what sort of habitat types, what sort of elevational gradients, all these different things might be contributing to selection for one fruit type over the other. So I hope that answers your question.
Yeah, no, I mean, I think you have to look at it, yeah, as you say, compared to other groups.
Right. The MELASTOMES, MELASTOMES provide a great model for beginning to tease apart these patterns, but it's, again, there's just so many other things that are just as hyperdiverse in these mountain ranges. So definitely good to keep an open mind with regards to what each clade is doing at each habitat type, right?
So related to that, Julie Denslow sends a question. What are your thoughts about the role of niche diversification in supporting these diversity hotspots?
Definitely a large component of these endemism hotspots, right? Because we know that this is still a very dynamic geological system. Hispaniola itself in particular, especially Southern Hispaniola, with orthographic uplift, with even on top of that, changes in climate during as recently as the Pleistocene and other mechanisms like that, it's safe to say that the vast majority of endemism and species richness that we see, especially in Southern Hispaniola, is a direct result of this habitat heterogeneity and these rain shadows, these cloud forests, these elevational transects, these gradients, these slices are clearly driving a large amount of this in situ diversification that we see.
Agnes?
Yeah, my question attaches perfectly to your answer now. So actually I have two, or a million, or 50, like Fabian.
Me too.
No, so I didn't really know about this this concept that species richness of small areas results from colonization of large areas and diversification in large areas and so on. But then if that is true for many groups of plants, I wonder whether you have any ideas or hypotheses why this may be different either for Hispaniola or for MELASTOMES. So I guess this probably will relate to your to the other part of your postdoc where you'll be looking at other clades too. And then the second question that I have is how you actually came up with the community matrices, whether you had inventories that sort of looked at communities and sampled all MELASTOMES, or whether you pooled occurrence data. It's just more a methodological question. So whatever you want to answer first.
To quickly answer that last one, we had Lucas Major had a pre-existing data set with the areas for each of these species. So it was relatively easy to kind of do a plug and play sort of thing with the phylogenies. Extracting locality data was a slightly more difficult task because ultimately what you can do is you can either, if you can't find locality data for one species, you could always just create like a centrum point for an area, for a biogeographic area, and just assign the species that occurs in that area that doesn't have any readily available locality data to that point. but that obviously is of zero relevance to any sort of endemism analysis. Thankfully, I was able to get good quality points for almost all of the species in the phylogeny.
That's pretty awesome.
Yeah, I know. Shout out to SciNet, shout out to GBIF, and other, large aggregating herbarium data sets, right? And then going back to your first question, which I've half forgotten by now.
Why you think, do you still remember?
Kind of. I remembered something about species richness in small areas, correct?
Exactly. And why do we see these patterns in melastomes if they are surprising in general? Or why do we see them in Hispaniola?
Well, going back to the previous answer I gave is, we have to kind of change, I guess, what we think about being a small area, because two-dimensionally it is, right? These are relatively small. This is a small peninsula in southern Hispaniola. But three-dimensionally, it's very complex, right? And then add on to that the effect of having rain shadows because of a trade wind, because of being on a windward or leeward side of a mountain. Then have, consider community assemblages along, different gradients along the mountainside, going from a low tropical to a kind of going from a sclerophyllous type vegetation to low tropical, just below cloud forest that has slightly larger leaves, going to a cloud forest that has macrophyllous leaves, plants that have macrophyllous leaves, and then going all the way back to these above cloud pine forests that then everyone goes back to these small narrow leaves. It seems that even if selection isn't immediately, at least notably, working at a genetic level, it's working at a phenotypic level, right? There's just a lot of phenotypic diversity along these gradients as well. And then when we look at the genetic data, it kind of corroborates this idea that it seems maybe one ancestor of a rapid radiation colonized a nascent mountain range in southern Hispaniola and kind of went from there, right? There are a lot of ecological factors at play, in addition to evolutionary processes such as in situ diversification and long distance dispersals of closely related taxa that, I don't know, melastomes maybe not, but in other groups might lead to some sort of hybridization. So it's kind of hard. I think the answer is there's a lot of different things going on here, right? And tropical systems are notoriously complex and have a lot of different factors at play. And that's what I ultimately hope to be able to tease apart, right, is by looking both at this genetic diversity and functional trait diversity, being able to kind of see, okay, well, what do we have here? Do we have clustering or do we have over-dispersion? And so far, it seems to me most of these mountain ranges are clustered, right?
So one question related to that is from Marcelo Reginato, and he asks, are there any obvious geological climate differences between the paleo and neo-endemic hotspots? And that makes me think that Central Cordillera, if I remember correctly, is a lot older than especially the things that are in the southern peninsula, like Mexico and
Yeah, exactly. A lot of times, I mean, even though, so it's kind of funny how they originally define neo and paleo endemism, because it instantly implies that there is a strong chronological definition to it, but in reality what neo and paleo is referring to is branch lengths, right? It's not looking so much,
but hopefully they are related,
right? Exactly. And so in this case, though, they match up perfectly. Newer mountains have more neo, older mountains have more paleo. And so that just, off the bat is is related obviously to this, the longer geological and climatological history of the Cordillera Central, which, if I'm not mistaken, Lucas, you can correct me if I'm wrong, is that part of Hispaniola was a continental fragment that kind of got lodged in between two plates. And then southern Hispaniola, and particularly the Tiburon Peninsula, is more of the product of this North American plate and Caribbean plate kind of uplifting and doing orthographic lift and other mechanisms like that. So yeah, from that, just from the, doing simply by reading the dates of the mountains, the ages of the mountains, and the geological history, it's the paleo and neo-endemism hotspots that we uncovered were corroborate with those natural histories of those mountains.
So related, well, a little bit different, but still on diversification, on these patterns and diversification process. Julie Denslow asked, how do recent arrivals, exotics, invasives, and I would add not only these exotics or invasive, but these things that we know are species that are widespread, like the Myconia crenatas of the world, or the Myconia dodecanthras that go from Cuba to Brazil, contribute to these patterns? Are they model for how they affect the models for diversification processes?
Well, they clearly affect how canopy and how biodiverse determine neo and paleo endemism centers as well. Because if you look at the community phylogenies for the Cordillera Central, a lot of times you see these more widely distributed taxa as occurring in that mountain range, as opposed to areas with a lot more strict neo-endemism, such as the Massif de la Haute, which is the most extreme example. So there's something going on there, and obviously the models are kind of picking up on that and flagging Cordillera Central as paleo, because it seems, like I mentioned before, it's a catchment basin for a lot of things from all over the place. With regards to diversification models, I'm not quite sure, but when we look at our diversity metrics of phylogenetic diversity and mean pairwise distance and mean nearest taxon distance, actually, I'm gonna go back to those. So it's interesting to point out here. Let's see, Cordillera Central C right here, and it's pretty low in comparison to Sierra Neiva, which is actually, that's something that I was kind of left thinking about a lot. And maybe Lucas has something to add with regards to how the Sierra Neiva compares in diversity to its neighboring mountain ranges. But it was kind of shocking for me to see such a disparity between what other metrics were recovering as very diverse regions, and then this one saying, no, these are not that diverse in terms of distance. Sierra Neiva seems to have the largest hodgepodge of different things from different areas, right? And least closely related.
Yeah, Andre, I guess one thing that I would say about that is, given its location, it does seem to be, this sort of mix and match of melastomes that are, so it has a lot of influence from both the Cordillera Central as well as Sierra de Bauruco, Massif de la Selle. And so, because it sits right there in the middle, it does seem to have this interesting mix of taxa from both of the other two major, relatively close Cordilleras. So potentially that could be what's driving this pattern that you're seeing here.
Yeah. So it's essentially a bridge, but at the same time, that bridge leads it to having kind of a less closely related community assemblage of melastomes. Yeah.
Any other comments, questions, complaints? So Walter
I think there was, oh yeah, you're asking Walter's question. Go ahead, Fabian.
Well, Walter just saying that it's interesting, that would be interesting to integrate these results with a largely missing information on reproductive biology on these MELASTOMEs. Yes, and I hopefully you can get a lot of that from your trait analysis. But I think that there are some of these things about, like, are they self-compatible, self-incompatible? Are they apomictic? Do they have one pollinator? Do they have 17 pollinators? We don't have the information for that for anybody. And you know, with hundreds of species in the Caribbean, 400 species, each of them is 1 or 2 or 3 seasons of study. This is not trivial data to have, but it would be awesome to understand, especially these things of self-compatibility and incompatibility, because some theories are that you want more self-compatible because if you have a small founder, you can establish yourself. But then why are there more dioecious plants in islands than off islands? Well, maybe because it's better to try to diversify yourself. So I think there are 2 opposing forces here, and see who wins. But with the data, it'd be a lot of data to get, a lot of work to get this data.
Right. Yeah, it's the competing forces between avoiding inbreeding, but at the same time also avoiding going extinct because there's nobody around you that's related to you. So yeah, very complex processes in reproductive biology that, again, we don't know anything about for this group, right?
Yeah. So if there is not anything else, I mean, this was awesome, and I think there's a lot more to come. I just want to remind everyone that in a month, in the 2nd week of October, I believe, we'll be announcing it soon. It's already actually on the website. We'll have Jeff Mancera from the Philippines talking about Asian MELASTOMEs. And then after that, we have talks on microLEC and maybe other things already lined up all the way to January. So again, if you feel that you want to talk, give a talk that is MELASTOME related, please, by all means, volunteer, send an email to anybody on the website or even just to the list. And I again want to reiterate that it doesn't have to be a flushed out data project. If you have an idea that you want to discuss with people and make it more like a chat, we're happy with that. And if you think that you don't have a full hour, then we'll combine 1 or 2 or 3 people with you to give like a mini symposium. We can know any and all of that. So encourage your students also to give talks. And this is a great space, and I'm learning a lot of every time I see one of these talks. So thank you very much.
Yeah, thank you, everyone. Looking forward to seeing you in October. And thank you, Andre. It was a super nice talk.
Thank you so much to everyone for having me again. This is a great group, and I hope you enjoyed my results.
We did.
We did.
Thank
Bye-bye.
you.